Good afternoon, everyone. Thank you for joining us for a short discussion on target translation with medicinal chemistry. And here with us is Dr. Thomas Keller. Can you briefly introduce yourself?
My name is Thomas Keller. I have a PhD in organic synthesis. Currently, I'm a consultant for EDDC in medicinal chemistry.
I keep encountering the importance of structure when it comes to medicinal chemistry. With the method of screening, with virtual or some other methods, if the structure is not completely revealed, it's a bad idea. And why is that?
As a medicinal chemist, you encounter thousands of structures, and if you have been in the game for 20, 30 years, you've worked on many different projects, and so you encounter a lot of different structures, from natural products to antisense molecules and so on. And so when you look at the structure, you can tell pretty quickly whether, for example, you've seen the structure before or the scaffold.
Right.
And usually what you try to assess is, does it have an obvious toxic liability? Are there some functional groups that are reactive, which would be a kind of a problem for a lead optimization? Also intercalators, compounds that are totally flat and that like to intercalate in the DNA. This is a toxicity problem. Right. So when you look at structures, you just try to see these obvious kind of red flags, or maybe you've worked in a similar class before, and you can already say, well, there's lots of work has been done in this class. Right. Finding a patentable position could be quite difficult.
What exactly is selectivity?
I mean, this has been changed with chemical biology now becoming really, really important. 20 years ago, as a medicinal chemist, we would have been very happy to optimize a non-selective compound basically on a phenotype, on a cellular assay, and just kind of try to get a very potent compound that does something that we want. These days, that would be very difficult because everybody wants to understand the mechanism of the compound. Everybody wants to know about target engagement in the cell. Does the compound really interact with the protein in the cell? Right. People want to know biomarkers for the clinical testing of the compound, and of course biomarkers of a compound that interacts with multiple proteins is very, very difficult because it's difficult to really pinpoint what the compound actually does to come up with the activity that you want. Yeah. So slowly in the last 20 years we have come more and more to a point where selectivity is really, really important, and understanding the mechanism by which the compound works in the disease area that you're targeting.
That's great. We're coming back to this virtual screening. There's more and more popularity for it. Every field of science is using more and more virtual ways. What are the pitfalls of virtual screening when it comes to selecting a compound as a good lead?
The pitfalls are basically the computational chemistry methods that are available. I mean, in principle, what you really would like to do is calculate the interaction from first principles and actually simulate a delta G of interaction, right? But that is not possible. And so what we are using is force fields, which simulate the interaction of the compound with the protein by using mechanical models. Right. And so these are not that accurate. And in the last few years, this has improved a lot. And so that's the reason why we get these false positive rates, right? That's often, depending on the druggability of the target, 50% of the compounds, when they go to a biochemical assay, they do not bind. I think it's becoming better and better. And so virtual screening is becoming a more attractive way to do lead finding. But I think the most important thing is that when you do virtual screening, you need to confirm the hits in a biochemical assay, ideally, or using biophysics, crystal structure, thermal fluor, ITC, or whatever, right? You just need to prove that your compound interacts with the protein. And of course, eventually, you should go to a cellular assay and really see whether it has a functional effect.
Let's say from a virtual screen of 200,000 compounds, and this group comes to you for advice. We have come up with these 100 molecules as real good hits. We should progress further. So what is your first advice? What should they do next before they say like, all right, let's take this compound a little further towards development?
There's been a big progress in the last 3-4 years. And so I would first advise that probably 200,000 compound is probably the lower limit of what you should screen, right?
Okay.
I mean, these days it's pretty easy to screen 10 million, 20 million compounds. It may be more expensive, but it's often worthwhile. And then I think what I would advise you is to visually inspect the poses in the crystal structure.
Yeah.
I mean, hopefully you've used the crystal structure, not a homology model for the virtual screening. A homology model works, but the false positive rate will be higher.
Right.
Crystal structures are the preferred thing to use for virtual screening. And then, as I said, I would like you to inspect the poses of the compound in the protein and see whether they make sense. And this has to either be done by a medicinal chemist or by a computational chemist. Best would be if both of them work together. And the next step then is to order the compound or to synthesize them by EDDC or by medicinal chemists, synthetic chemists, and then test them either by biophysics or in the biochemical assay. And the final step is then, if they're really potent enough, you can try them in the cellular assay.
It's a super naive question, but when you guys say any structure you guys can just synthesize in your lab, do things ever explode in your lab while synthesizing new molecules that you have never done before? Or like, is it all pretty proved?
I mean, that's the thing of medicinal chemist. We always synthesize molecules that have never been synthesized on this planet, right? That's why we can patent it. That's actually what people often don't understand. They may exist virtually, but they have not been synthesized by anybody.
Right.
And so that's what medicinal chemists do. We try to come up with new molecules for the target so we can patent them. If somebody else has synthesized them before or used them before, getting a patent is quite challenging.
What is one of the surprises that you've gotten while synthesizing a completely new molecule that never physically existed?
I mean, unexpected surprises are
Is
always happening, right? Because what you're doing when you do structure-activity relationships, you synthesize molecule, you design them, and then you give them for testing. And when the results come back, usually there are some surprises that you didn't anticipate. I mean, the best design sometimes is totally useless. You synthesized or you designed a fantastic compound. I thought, oh, this compound is it. And it's totally inactive, which means that your model that you used for designing your compound was probably not correct. So during the whole structure activity development, you're basically trying to understand what the binding site of the protein is like. And even if you have a crystal structure, quite often the protein moves, right? We have conformational flexibility.
Right.
And so even the best design with the crystal structure sometimes gives you surprises that actually the protein has moved and, a residue that you wanted to interact with has suddenly moved an angstrom away.
Right.
And then, of course, the compound doesn't work.
Right. When you receive a compound for evaluation, what do you look out for? What's your process?
The goal of a medicinal chemistry evaluation is always to understand how expensive and how difficult an optimization towards a preclinical development compound will be. And when we think about that, I generally want to know three things. First of all, I would like to understand how was the compound identified? What lead finding method did you use to identify the compound? The second thing that I really want to know is I want to look at the structure. I want to see whether you have structure-activity relationships. And the third thing, which is a little bit more recent, I want to know what is the selectivity of the compound. And have you thought about looking at the selectivity in the future drug discovery phase. And of course, you may not have answers for all of these three questions, but I just want to understand whether you have thought about that and whether there's a plan.
A couple of things you said, one with the selectivity of the compound and the method of how we arrived at the compound for evaluation. Why is the method that they choose to evaluate a compound so important?
Hit finding, depending on what stage you are, has its weaknesses. And so I would like to understand, what have you done to confirm the activity of the compound? And for example, if you have done the virtual screening, you have used computational chemistry to measure, to understand the interaction of the compound with the protein. We know that the false positive rate of such a virtual screening is about 50%. And so I really would like to know, what have you done to confirm the activity in a physical assay? Have you done a biochemical assay, or have you done a cellular assay? And how sure are you that this is really a promising compound? If you have done an HDS, then we have to be careful of false positives because of aggregation. If compounds don't have very good selectivity, they often aggregate in the assay, and this cannot be seen by visual inspection. And so also in this case, you need to confirm the activity, and mostly in the HDS, you use biophysics to confirm the interaction of the compound with the protein. And for phenotypic screening, we always worry about the selectivity. We want to know, does your compound interact with multiple targets inside the cell, or is it really selective for one compound? Because that will change totally the way we look at the compound. And the most important thing overall is, how potent is your compound in the assay? Is it micromolar? Is it nanomolar? And usually, as a medicinal chemist, I would say, if the compound is below 10 micromolar, we can do something. If it's above 10 micromolar, we would be quite skeptical that this is a good starting point for a lead optimization.
When you decide these 10 micromolar and below is a good starting point, what happens if the affinity is more than 10 micromolar? How would it disqualify a molecule?
The issue with compounds that have an IC50 of greater than 10 micromolar is basically with the way medicinal chemists optimize the compound. We usually use structure-activity relationships to optimize a compound. And structure-activity relationships, by 30 years of experience in the whole drug discovery, from Pfizer, from Novartis, from all different companies, we know that structure-activity above 10 micromolar is not reliable. And the more potent the compound is, the better is the structure activity. So the optimization of a compound with an IC50 of greater than 10 micromolar is quite challenging, because the structure activity is not reliable.
Right. Since we're talking about the starting point, like a final product, a drug that is already in the market, at what, is it like working at a nanomolar concentration or a picomolar concentration? So what is the concentration at the end of the development?
As a final, usually the interaction with the protein would like to be in the nanomolar range. Of course, because of, for example, protein binding, the cellular activity will probably be about a little bit higher. And so I think for a cellular activity, we'll be happy with about 100 nanomolar. But on the protein, the interaction, the KD, the IC50, would like to have in the nanomolar range.
Dr. Keller, thank you so much for your time this afternoon, telling us about what goes into drug discovery and compound evaluation.
My pleasure.
And also thank everybody for joining us.